two main areas in the lab. One, which is the topic of today's talk, which is the genetic control of growth and metabolism, and another, which is developmental epigenetics. And there's, of course, crosstalk between these areas. But I would like to start by paying tribute to collaborative research. And for that, I'm actually using the famous quote of John Donne, which was a quite famous English poet that lived in the 17th century, that said that no man is an island. And I guess that's quite a powerful message, in particular in the days that we are all facing. And it's a good thing to remember that science is we don't work in isolation, it's very collaborative. And as such, I would like to pay tribute to a number of excellent collaborations that I have set up. These are the ones that are actually active. And in particular, in the context of today's talk, I would like to acknowledge the contributions of Cristina Branco, that is now in Belfast, the university, and Amanda Perry at the University of Cambridge, a colleague of mine. And there's, of course, other very important contributors to our work that help shape the research that we do. And for that, I'm really grateful. So the interests, I'm really fascinated by this maternal-fetal communication. And when the fetal communication goes wrong, what are the consequences in terms of pregnancy complications, from intrauterine growth restriction to gestational diabetes, preeclampsia, etc. And so we all know that at the very center of this communication is the placenta. And the placenta is worth remembering that is in essence a fetal organ. But it's a fetal organ that is continuously being bombarded by fetal signals emanating from the fetus and also signals that come from the mother. And we're really interested in characterizing what these fetal demands and maternal signals are. In particular, we are interested to understand the role of the fetal genotype and the maternal genotype. And so we use genetic tools. In particular, we use mouse models to try to understand these crosstalk between the fetus and the placenta, and then the placenta to the mother, which is really a complicated and complex system. And so we focus actually on the insulin-like growth pathway. And we have done some work also with Amanda on the PI3 kinases. But so today, what I would like to do is to answer two questions. I may only answer one because we may run out of time, but then Alejandro will tell me if it's okay to continue or if I should stop there. And the question that I really would like to address is this one: how is fetal growth coordinated with placental growth and function? And provided we have time, how do fetal genes influence the maternal metabolism? So we're all talking about here about the fetal genotype and maternal metabolism as the question number two. And I don't think I'll have time to go through the work that we have done. This has been published a few years back. So in my mind, it will be more interesting for you to learn of new stories that are going on, which is this: do fetal and maternal genomes interact at the level of placenta to modify maternal resource allocation? So as I said, we use the insulin IGF signaling system as a model, as the pathway of choice. And you all know much better than I do that postnatal growth is mainly controlled by the growth hormone and the IGF-1 axis, and very important work done by E.F. Stradiatis on this system has actually tell us that the growth hormone and IGF-1, they probably can explain about 80 to 90%, probably more, 90% of all the growth postnatally, which is still, we still have about 11% to go and fish out what are those main pathways or other alternative pathways. But in terms of so that's postnatally. In terms of the embryo, in the fetus, there are, of course, many pathways that are important for fetal growth, but one of the ones that are really highly conserved and quite important is the insulin IGF signaling, as you well know. And we have to input not only these growth factors, but also the placenta. And the placenta is totally crucial here as a component when talking about growth in utero. What really I find fascinating as well is that there are two genes within this pathway, IGF2 and IGF type 2 receptor, that are actually regulated by epigenetic mechanisms in a process that we call genomic imprinting. So genomic imprinting is the expression of genes according to parental of origin, and you have IGF2 being a gene that expressed from the paternal allele, and IGF only, and IGF type 2 receptor is expressed from the maternally inherited allele only. So this is a paternally expressed gene, this is a maternally expressed gene. And so these or at least IGF2 actually is located within a cluster. So here it is. And this cluster is called the imprinting growth cluster. And why do we call it imprinting growth cluster is because there are a number of genes here that are quite important for placental function and development and growth, and also key genes that are involved in growth. So there are two human syndromes, the Beckwith-Wiedemann syndrome and the Silver-Russell syndrome, that are what we call imprinting disorders, and they are the result of these imprinted genes going wrong, the expression of them going wrong, and I'll get back to that in a minute. And so the IGF2 is part of what we call the ICR1 cluster. So ICR stands for imprinting center region, or imprinting control region. And there is another very close by, another imprinting control region, too, with many more imprinted genes, as you can see here. And so this is quite fascinating because these imprinting control regions, they actually control the expression and the imprints of these genes. So these genes are controlled by ICR1, these genes are controlled by ICR2. And Beckwith-Wiedemann can result from problems in ICR1 and ICR2, whereas Silver-Russell so far has described problems in ICR1. And I will explain a little bit more in detail how this works, because it's quite important then to understand the rest of the story that I have to tell you. So we're just focusing now on this region here, which is ICR1, where IGF2 is located. And so this also is good to explain basic principles of genomic imprinting, which is a remarkable biological mechanism. So here we have the IGF2. Everybody knows this is insulin-like growth factor 2 gene. And downstream of IGF2, there is this gene called H19, which is a non-coding RNA. And so they are controlled by this ICR1, which is here. And I'm going to explain to you how this actually works. So as I've told you, IGF2 is a paternally expressed gene. So that's indicated here by this arrow. So it's expressed when it's inherited from the paternal allele, and therefore the maternal allele is silenced of IGF2. So it's a paternally expressed gene. Now let's also discuss why we're going to discuss why there is this silencing on the maternal allele of IGF2. But before, I'd like to ask you to pay attention to this gene here, H19, which is a non-coding RNA, and as reciprocal imprinting. So these are only 90 kb away, and nonetheless, H19 is a maternally expressed gene, and the paternal copy is going to be silenced. So IGF2 paternally expressed, H19 maternally expressed. So that means that the other alleles in both will be regulated by epigenetic mechanisms. So going back to that question that I told you, so let's see how IGF2 is silenced specifically on the maternal allele. And this is because of this ICR1. So ICR, the imprinting control region, is a sequence of DNA and just located upstream of the H19 gene. And when this region is unmethylated, so the methylation of DNA, as you know, is the addition of methyl groups to the DNA, and you can have methylated DNA or unmethylated DNA. When it's unmethylated, this means that there is a insulator protein called CTCF, represented here, that can bind because this region is unmethylated. And what CTCF does is it does not allow these enances that are located downstream of H19 to loop around and contact the IGF2 promoter. So both IGF2 and H19, they require the action, the proximity of these enances for full activation. And so CTCF is here. This is like a barrier, doesn't allow these enances to communicate to upstream IGF2 promoters, and therefore IGF2 is completely silent. But on the paternal allele, the story is slightly different because this ICR1 now becomes methylated. And this methylation is a germline methylation. So we know that the sperm comes methylated and the oocyte comes unmethylated. And soon after fertilization, this DNA methylation actually goes across the gene, the H19 gene, and does not allow the H19 to be expressed. And of course, the reason why H19 is expressed on the maternal allele is because this region is unmethylated, and therefore the enances can freely contact with the promoters, but they cannot go further because of the CTCF. And so the methylation means that CTCF on this allele cannot bind, and therefore you get the enances contacting IGF2 promoters, and that's why you have paternal expression of IGF2. So this is the mechanism that we call the regulation of an imprinted cluster through a CTCF-dependent boundary. And why is this relevant is because there is this concept of epimutations, which I find really remarkable. And these are work mainly done in France by Irene Nachin, and also at the time Boss, which they reported the following in terms of the Silver-Russell syndrome, and this was work done by others in terms of the Beckwith-Wiedemann syndrome. So this is just here, I'm just showing what I said to you before. So that's the IGF2 allele here. And so the IGF2 gene here, which is paternally expressed, and I'm not representing here the H19 for simplicity. So this is the normal situation. CTCF binds the unmethylated ICR, doesn't allow the maternal allele of IGF2 to be expressed. But, and this is the beauty of the system, if you have an epimutation and there is the gain of methylation, of DNA methylation specifically on the maternal allele at this ICR1, we call this gain of methylation. So CTCF can no longer bind, and this means that you have biallelic expression of IGF2. And if you have too much IGF2 in early development, or double the dose of IGF2, you will get this overgrowth syndrome, which is a rather rare disease. You probably are very well aware of it. So the Beckwith-Wiedemann is probably 1 in every 12,000. And this is an overgrowth syndrome because there is excess of IGF2. And so this explains this gain of methylation. Epimutations can explain about 10% of the cases with them. Vast majority of epimutations explaining Beckwith actually occurring at the ICR2, which involves a non-coding RNA. Nonetheless, there are other mechanisms by which you can have Beckwith-Wiedemann, which is literally the same as an epimutation. Is if you make a genetic deletion or microdeletions that have been reported in the literature that have exactly the same effect. And then this extraordinary finding that you can also have within the same ICR1 the Silver-Russell syndrome, which is characterized by this very severe growth restriction phenotype. And in this particular case, what you have is loss of methylation on the paternal allele. So you lose the methylation, therefore CTCF can bind partially here, and you have less IGF2, and this gives rise to Silver-Russell syndrome. And these are P mutations. This is loss of methylation has been found in about 60% of the Silver-Russell syndrome cases. So a little bit about what we know about the IGF2. IGF2 can bind to these receptors here, the IGF type 1 receptor, but also to the insulin receptor and to the hybrid receptor. And when it binds to these, it will initiate a cascade, classical pathways from two classical pathways, which is PI3 kinase and the RAS. And these can explain most of the metabolic and cell proliferation and anti-apoptotic effects of IGF2. Of great interest as well is this receptor here called the IGF type 2 receptor, which binds IGF2, but also binds other molecules, such as proliferin and the leukemia inhibition factor. And for many years, we have seen IGF2R, mainly seen IGF2R as a clearance receptor for IGF2. What does this mean? It means when IGF2 binds here, there's going to be cleared by the endosome and degraded through to the lysosome, and therefore you get rid of excess of IGF2 in the extracellular milieu. And so that is the canonical function of IGF2R, and there's always a bit of controversy in the literature if this receptor actually has any signaling properties. But new data has actually shown that there probably are some signaling properties through the G proteins. So you can have signaling through the G proteins and the phospholipase C, and this has been seen in the context of neuronal plasticity. And most recently as well, the evidence that they can actually link up with the ERKs. And so this is something to bear in mind when I discuss our results. And so genomic imprinting is this amazing parent-specific marking of genes that occurs in the germline. And then you have this differential expression depending if it comes from the mother or the father. And so people see there's one theory of evolution of imprinting that sees this as an arms race. And this is the canonical, the paradigm example. Here you have IGF2, which promotes growth, and it's a paternally expressed gene. So you see expressed from the paternal allele, the maternal allele is switched off. So this is a genetic arms race for what? So it's a parental tug of war in relation of allocation of resources. So paternally expressed genes in the offspring serve the interests of the paternal genome, and they are selfish and they're very greedy, and they tend to get as many nutrients from the mother as possible so that the offspring of those paternal alleles can be passed on in the population pool. And of course, the interests of the maternal genome are going to be the opposite, antagonic, in the sense that the maternal genome is interested in distributing all the resources that she has evenly to the offspring. And of course, she wants to be able to reproduce again and again. And so the response of the maternal genome to this greediness of the paternal expressed genes through IGF2 is to come up with the IGF type 2 receptor, which what it does is actually clear the excess of IGF2 in circulation. And this, amazingly, is a maternally expressed gene. And so this acts at the level of the protein, but also at the level of transcription. Now, IGF2R is maternally expressed only because this is a growth suppressor, and the paternal allele is going to be silenced. So there is this amazing arms race going on in our genomes, according to this theory. And so going to the topic of this talk, so this is the question I would like to give an answer today. And this is really about how does the fetal growth coordinates with the placental growth and function. And you'll see the questions that we're addressing here. And so this work was mainly done by Ionel Sadovici in my lab, helped with a master's student. So he's a very talented, as everybody says, uses the word talented. I think he is more than talented. And I, Katerina Georgopoulou, which was a master's student working with us. And so this work is under review at the moment, and you can find some details in an old version in bioarchives if you're interested in knowing more details about this story. So I will have to give you a small introduction about the mouse placenta, because we're using genetic models to understand how this is coordinated. And so the mouse placenta seems very dissimilar from the human placenta if you look at it, but many cell types are actually conserved and many of the molecular mechanisms are conserved. So that's why we don't have a problem in using the mouse placenta as a model. But of course, there are many other important differences that need to be taken into account. But in terms of the mouse placenta, there's three main layers. So the maternal layer, also called the sedua, which is shown here. And there are then the spongiotrophoblast layer or junctional layer, which is this layer here, which is composed of trophoblast giant cells, glycogen cells, and spongiotrophoblasts. And this is what is amazing about the mouse placenta, because it's a great tool for us, is that this is the endocrine layer. So most of the endocrine output from the placenta in the mouse comes from this layer. And we can genetically manipulate this layer, so we can actually make some strong conclusions about the endocrine output of the placenta to the mother and others that alter maternal metabolism. And this is something, this layer does not exist in human placenta. So in the human placenta, the endocrine output is done by the transporting epithelial as well. So it has the dual function. But the most important, equally important, let's put it like that, is this, because of course that you don't have placental development if you don't have this spongiotrophoblast layer. The labyrinthine layer is the one where you have the exchanges of nutrients from the mother to the fetus and gases, et cetera, and waste. And so this is composed, it's what's zoomed here, composed by the following cell types, which I will be referring to, so it's important that you remember some of this. So we all know that the maternal blood does not mix with the fetal blood because there is this interhemal membrane here that doesn't allow for that. And there's two layers of syncytium. And so these two layers, layer 1 and layer 2, with layer 2 closest to the fetal capillaries. And so they are, as I say, they are syncytium. And so the nutrients need to go across this membrane here, this exchange barrier. And then you have also these, what we call the sinusoidal trophoblast giant cells as another layer, which actually also has some endocrine output into the mother also, we think, in those terms. So when we talk about the fetal capillaries, we'll be talking about this area here, the maternal blood spaces. And when we talk about the trophoblast, we're talking about these, mainly these two layers of syncytium. And what has been great is that we have Cree lines, and so this is part of the way we can genetically engineer these layers. We have Cree lines that we can cross with conditional knockouts that have the gene of interest with 2 LOXP sites in between that gene of interest, and that can be deleted through the action of this Cree recombinase. And this is an example of a beautiful one, which is called TVP-A-Cree, generated by Gustavo Leone and Jay Cross's lab. And you can see here, this is the reporter marker. You can clearly see that this is green, and this actually corresponds to this layer here. So this is a Cree line that is expressed specifically in the endocrine layer of the placenta. And so this is more or less what I would like to say as introduction. So what we're really trying to understand is how does the placenta supply matches to the fetal demand for growth and for nutrients. So you have to have, so the supply is represented by the placenta, the demand is represented by the growth of the fetus. And so you need to have a perfect equilibrium between supply and demand. In other words, as this fetus grows, it needs to be given enough supplies through the placenta. And so this is a balanced system of supply and demand. But one of the things that really fascinated us for quite a while is this adaptation that the placenta does in late gestation, which is depending on the species. And this is the importance of this type of work, and I guess why we elect doing mouse genetics, because in every single species that has a placenta, the surface area for nutrient exchange is going to be increased 5 to 15-fold between mid and late gestation. And so this is because as you go along with gestation, the fetus grows and needs more food, and therefore there is an adaptation of the placenta to give more food, and it does that by increasing the surface area. And a key element of increasing the surface area is this expansion of the placental vascular tree represented here for the mouse. So there's a continuous expansion of the fetal placental microvasculature. And this is always associated with what we call morphogenic modifications in the overlying trophoblast epithelium, the situ that I've talked to you about. So they go hand to hand. And we know that there are many knockouts that if you mess around with the overlying trophoblast epithelium with this in situ, this has an impact on the vasculature. But the crosstalk between the vasculature and the trophoblast is yet pretty much unknown. So that's much less known if you mess around with the vasculature, what happens to the trophoblast. And so this is one of the key questions that we're addressing here. We want to know why in all placental mammals there is this expansion. Well, we know why this is, but how does this actually occur? And the way we do it is by manipulating the size of the fetus independent of the size of the placenta. So we've done we've published a number of papers where we were able to manipulate genetically the size of the placenta. And so this is a small placenta, and then we ask the question, so specifically in the placenta, and then ask the question, what happens to the fetus? And what are the crosstalk that exists between the two? But one thing that we hadn't done yet, and this is what I'm going to talk to you about, is one that actually makes the fetus smaller and then asks the question what the impact on the placenta this might be. And so that's the work by Yunel and Katerina. And we actually broadened this from IGF2 to the IGF signaling system. And it's a lot of work that involves about 7 genetic models. So for us to be able to conclude some important things. So as I told you here, so we have this expansion of the fetal placental vasculature, the placental tree. And so we had to look at this in the mouse. We already knew that it's very likely, but nobody has done this characterization. So this is the fetal weight, and this is gestational age. And you can clearly see that most of the fetal weight gain occurs between 16 and 19 when the fetus acquires most of the mass, really. It's in very late gestation. And the placenta in the mouse, the peak of growth is day 16, and then it actually plateaus from 16 to 19. And these are the different layers, the chorionic plate, the labyrinthine. So remember, this is the exchange barrier where the maternal nutrients cross to the fetus, the junctional zone, which is the endocrine layer, and the maternal decidua. And so you can see that in spite the placental weight actually grows and then stops growing, the labyrinthine, which is where the vasculature is, actually grows throughout gestation. So this stop is not because of the expansion of the vasculature that continues throughout gestation. And we did more. We then correlated the labyrinthine weight, which contains the vasculature, with the fetal weight, and we could see a very good correlation, in particular at T19, where the fetus is biggest. That is a very strong correlation between the labyrinthine weight and the fetal weight. So there is this continuous expansion, as I explained. And so this raises the question that maybe there are signals that come from the fetus that participate in this expansion. And this is what we wanted to address. Are there any fetal signals? And if there are, what are they? And so one of the observations we did, because we've been interested in IGF2 for quite a while, is that we finally got a way of measuring the levels of IGF2 in the fetus. And from day 16 to E19, as you can see here, is almost a double of the amount of IGF2 in circulation. So IGF2 can act as a normal-like protein, as you well know. And then we also did a correlation of the IGF2 with fetal weight, and there is a very reasonable correlation. So the more IGF2 you have in circulation, the bigger the fetal weight. And this, I know that there's some controversy about this in humans, with some papers saying yes, other papers saying no. So given this results here, we thought, oh, maybe it's IGF2 itself that may be having some role in the expansion of the placental vascular tree. But one thing that I should say is that IGF2 in the mouse is expressed widely in the fetal tissues and also in the placenta. So if you take IGF2 from the fetus and the placenta in what we call a total null, you have about a mouse that is 50% smaller and stays smaller, but otherwise is a kind of a mini mouse that stays smaller and is fertile. But we know it's very highly expressed in fetal tissues and also in the placenta. And so then we had to look where IGF2 exactly is expressed in the placenta. We knew that is widely expressed in the entire placenta, but where precisely? And we were taken aback by these findings, is that when we looked at IGF2 mRNA, it looked like, so this is an in situ hybridization. So if you look, it looks like it's on the fetal capillaries and the endothelial cells here. And then we went on to perform immunohistochemistry, so to look at the protein. And indeed, these spots you can see with the highest expression are from the endothelial cells and the fetal capillaries, with the trophoblast, in situ trophoblast expressing much less than in the endothelial cells. That can you clearly see here the quantification. And when we did an RNA-seq specifically on these endothelial cells, it was a big surprise to see that IGF2 is #1 gene expressed in these endothelial cells. So remember, these are endothelial cells in the placenta. So they are the feto-placental endothelial cells, and expresses the gene that is expressed the most is IGF2. And as another remark, it's amazing that there's a number of other imprinted genes that are actually highly expressed within the first 100 genes, highly expressed in the endothelial cells. Maybe imprinted genes have particular roles in these endothelial cells. So given this, what we then proceeded to do is to analyze this genetically. So Unell had made a conditional knockout mouse. So this is the flox alleles. So when you cross these mice with specific Cre lines that express in specific cell types, these two flox P sites are going to be deleted, and you end up by having no IGF2 being produced. So we wanted to make a mouse that was smaller. So we wanted to make a mouse that does not have IGF2 in the fetal organs. And we can do that. So this is, when you see this diagram, it will always be referring to the fetus-derived IGF2 knockout. And so what we did was to cross these mice with MEOX2 Cre, and we call this the epiblast knockout because it takes IGF2 from the epiblast, and the epiblast is what gives rise to the fetal organs. So we've made a mouse that has no IGF2 in the fetal organs, but remember that the fetal capillaries and endothelial cells come from the fetus, so they do not come from the extraembryonic tissues. So this particular mouse model, which we call fetus-derived IGF2 knockout, takes out IGF2 in the fetal organs and in the fetal placental endothelium. And this is important for what we're going to see next. So when we looked at what is the effect of this in terms of fetal weight, it's no surprise that if you take out IGF2 from fetal tissues, the fetus will be very growth restricted. And this is the mutants versus the wild types here. So you can clearly see that they're about 40% smaller than the corresponding wild type littermates. I guess the surprise was to see that there is a profound effect on the placenta. So you're taking out the fetal IGF2 in the organs, but also in the fetal placenta endothelium, and you have this huge impact on the placental weight. And so, of course, because we know that IGF2 is expressed highly in the endothelial cells of the placenta, we then did a knockout, which we call endothelial cell IGF2 specific knockout, which takes out IGF2 from the endothelial cells in the fetus and in the placenta. Fortunately, we don't have a placental-specific endothelial cell which we could interrogate. And what you can clearly see here is that there is an impact in the fetal weight, but it's a much smaller impact than you see when you take out all of IGF2 from the fetal organs. And then the placental weight is the same. So it's you do see an impact on the placenta, but it's quite different from the one that has more IGF2 being knocked out. And when we looked at what happens to the expansion of the labyrinthine that I talked to you about, these are the results. So you can see here, so these are the controls. And you can see, so this measures all the layers of the murine placenta with the labyrinthine, junctional, and maternal decidua. And you can clearly see, just looking at the controls, that there is the expansion in volume I've talked about, which correlates with weight of the labyrinthine from E14 to E16, and then from E16 to E19. But if you have a look at the knockout, you can see that it's completely blunted. So the expansion is not occurring anymore. And it doesn't occur as well as it should in the endothelial cell IGF2 knockout either, but which with the phenotype is much less prevalent in the endothelial cell IGF2. And so this led us to conclude that the fetal and endothelial IGF2 are main controllers of this placental labyrinthine expansion. So you need fetal IGF2 and endothelial IGF2 to have this expansion of the labyrinthine. And to look a little bit more in detail, what Yonel did next was, okay, so if this is a problem with the labyrinthine expansion, maybe what we have is a problem of the trophoblast and or of the endothelial cells, the fetal capillaries. And he micro-dissected the labyrinthine zone, the exchange barrier, and he did an array. This is still the days of arrays. And you could clearly see that angiogenesis and vasculogenesis were the very top enriched pathways. So which to us tells us that there's something wrong with the vasculature in these mice. And he was able to detect a molecular signature involving angiopoietins. So these angiopoietins bind to the TEC receptor, and they control endothelial cell survival and proliferation. And you could see that in our mutants, you see profound reductions of the angiopoietin and also of its receptor at the late gestation, where we see this defect and upregulation of the angiopoietin 2, which is the inhibitor of this of the TEC. And so when we looked at the fetal capillary volume, you could also see that there is a dramatic loss of the fetal capillaries in these mutants, which is in agreement with these molecular defects. I'm not saying that these are the culprits for this, but certainly they would have contributed. But this is not simply the vasculature that is affected, because as I said at the beginning, the vasculature is always normally correlated with the effects on the underlying epithelium. And we see that the labyrinthine trophoblast here is actually also affected in terms of volume. So in other words, you have less syncytium being made, the barrier that I talked to you about. And it could find that there were drastic reductions in genes that are very important for differentiation of the syncytium. GCM1 and SYNB are really downregulated in the mouse mutants compared to the controls, which suggests that this is impacting on the morphogenesis. And therefore, the conclusion of this is that if fetal-derived IGF2 is going to be is essential for this expansion of the microvasculature, or at least this is how we saw it at that moment in time. And so we then wanted to zoom in about what are the molecular defects that we actually see in addition to these, and we've measured apoptosis, and you can clearly see that there is a very increased apoptosis at E16 in these mice, but also a big reduction in cell proliferation at particular, always at E16. So we know that we're losing some of the endothelial cells because there's increased apoptosis and there is less proliferation, probably driven by a combination of circulating IGF2 and endothelial IGF2. And if you don't like these graphs, you probably will appreciate that it's pretty convincing this. So this is the control and this is the mutant. And at E14, there is no differences because we see actually that the growth of the placenta is much less affected at E14. But when we go at E16 and E19, term is E20 in the mouse, you can clearly see there is a big loss of, so this is a marker, immunohistochemistry marker for the endothelial cells. So it's a CD31 marker. And you can clearly see there is a loss of these endothelial cells that actually is disproportional to the other cell types. So we have a profound impact on the fetal capillaries and endothelial cells. And we went further to kind of count or quantify this defect that we're seeing. And this is represented here, which I find is totally amazing. So these are actually the total number of endothelial cells by flow cytometry. And you can see, as we have been mentioning time and time again, that there is this expansion. As you go along from E14, 16, and 19, you have an increased number of endothelial cells because they are, of course, part of the placental vasculature and they need to be there to meet the fetal demands for growth. But if you then see the mutant, this is completely blunted there, very little. But of course, these mice already have a small placenta at E16 and E19, so it's proper that we normalize this in terms of the percentage to other cell types, because the placenta is obviously smaller. But what you can clearly see here is the evidence that these are disproportionate losses of the endothelial cells in the mutants that lack fetal IGF2 in their organs and also in the fetal placental endothelium. And so the labyrinthine weight completely lacks that expansion. And this is very nicely correlated with a major drop of IGF2 in the circulating in blood. And then when we look at the endothelial cell, which I already told you has a much less impactful phenotype, this is the one again that takes out IGF2 from the endothelial cells in the fetus, but also in the fetal placental endothelium. We see a reduction much later on in gestation. We don't see a disproportionate loss. So the impact of this seems to be circulating IGF2 because you have much bigger loss here than you see here. So there's a contribution of the circulating IGF2 in kind of stimulating the growth of the endothelial cells and the placental capillaries. And again, the evidence that there is a blunting of the expansion, and this is associated with a much smaller reduction in the circulating IGF2. So it looks like the expansion of the fetal placental microvasculature requires both the endocrine, which is the circulating IGF2, and the paracrine signaling, which is given by the endothelial IGF2 or even autocrine signaling in endothelial cells. And so we went then further to study what happens if you take out IGF2 from the trophoblast. So this is taking out, so here we take out in the fetus and in the fetal capillaries. Here we're just taking out in the trophoblast, in the syncytium, and in the other layers of the placenta. So this is the extra-embryonic tissues. And we had another model, which is the H19DMD, which overexpresses IGF2. So this is a gain of function of IGF2. You have double the dose, a little bit less than that, but you have an increased dose of IGF2 both in the fetus and in the placenta, and in the fetal capillaries of the placenta. So what do we get? We get here what I've already shown you for the epiblast knockout. But when you take IGF2 from the trophoblast, nothing at all happens to the fetal capillary. So what IGF2 is doing in the trophoblast, in the syncytium, which is at lower levels than endothelium, is not impacting on the vasculature at all. And as we would expect with a gain of function, you can see that the total number of endothelial cells increases quite dramatically. And this all associates beautifully with the levels of IGF2 in circulation. So the most impact on the fetal capillaries is when you have loss of IGF2, dramatic loss. There is no changes in fetal circulating in the trophoblast deletion, so there's no impact on the fetal capillaries. And then there is an increase of IGF2 in circulation, and this is associated with bigger numbers of the endothelial cells. In the last few slides, just to tell you that we then were chasing what might be the receptor that this is all going through. And Unal made this kind of difficult experiment, which was to isolate placental endothelial cells from a mouse at E16, and he cultured them in vitro. And he found that, actually we knew from in vivo, but he confirmed that ex vivo, the receptor that seems to be mostly highly expressed is actually IGF2R type 2 receptor. IGF type 1 receptor is also expressed at very good levels, but there is no evidence that the insulin receptor is at all expressing in vivo in the endothelial cells. And this, of course, was in fact sorted cells. But in this cellular system of ex vivo, you could recapitulate the findings in vivo. So you could see that IGF2R type 2 receptor is the highest, much lower IGF type 1 receptor, and very little of the insulin receptor. It was surprising, though, that when he cultured the cells, the levels of IGF2 in these endothelial cells went pretty much nothing. After a few days of culture, IGF2 switches off. And this actually gave us the opportunity, because there's no endogenous IGF2 being produced, to make an experiment, which was to culture these cells in presence of IGF2. So this is the controls. So if you put IGF2 in these cells, they start making these capillary-like tube structures, which is indicative of angiogenesis. And so that was quite good. And then when Unel used an analog that goes through the IGF type 2 receptor only, you could see that there was also ability of making these capillary-like tube structures, but to a much lesser extent than the IGF2 only. And he went a step further, which was to use this inhibitor of the IGF type 1 receptor pathway, which is PPP, picropodophyllin, I think that's the name of it. And when he put the inhibitor together with IGF2, he could see that there was more or less the same growth of these tubular structures as the IGF2 was. So meaning if you inhibit IGF1R, nothing is happening really. So we pretty much convinced that this is going through IGF2R and not IGF1R. This is in vitro, but also have some in vivo stuff to show you. And so we then use these same cell lines this were really in response to the reviewers' comments, to do an siRNA to the IGF type 2 receptor. So in these same in cells that were actually bought from a company, they are a little bit at an earlier stage, but we were able, or Vicente was able to do this cool experiment, which was to do the siRNA for the type 2 receptor. And you could see that the siRNA was working beautifully. And then he did a cell proliferation measurement. So you can see here that's the IGF2R siRNA, and this is the scrambled, and there's a clearly significant defect on proliferation when you have the siRNA. It's a modest one, but nonetheless. So this means that there must be IGF2 independent effects of the receptor, because this is independent of IGF2 being there or not. And then he went on to do the scramble siRNA plus IGF2, and you can clearly see that this really activates the proliferation of these endothelial cells, as you probably would expect. But the biggest surprise was that when you do the siRNA in the presence of IGF2, you get a much bigger difference in downregulation of cell proliferation that you saw without IGF2. And so this was quantified, and there's clearly IGF2 independent effects. IGF2R effects that are IGF2 independent, and those that are actually dependent on the IGF2. And he went on to look at possible molecular downstream targets. And to cut the story short, he actually saw, so this is an acute stimulation of IGF2 in these cells, and he could actually see that there is a significant delay. So this is the scrambled siRNA, and this is the siRNA for IGF2R in the presence of IGF2. And he could clearly see there is a significant, so this is the control, significant delay in reduction in the ERK-1,2 phosphorylation, which is quantified here. So we think that the IGF2 dependent effects are going through the ERK pathway. And lastly, in terms of in vivo, we also have made a knockout of the endothelial cell for IGF receptor type 1. And as all the data that we were being collating, there was no evidence from all the data that this was going through IGF type 1 receptor. And this seems to be the case, because when you take out IGF type 1 receptor from the endothelial cells, there's absolutely no effect on fetal weight, placental weight, labyrinthine weight. And there's, if anything, an increase in the number of proportion of endothelial cells. But when we did this IGF2R specific knockout in the endothelial cell, remember that IGF2R is also a clearance receptor. So because it's a clearance receptor for IGF2, this is associated with an increased level of IGF2 in circulation, and the placentas are actually bigger. But when we measured the amount, the number of proportional number of the endothelial cells, we saw very much a similar pattern to what we had seen for the loss of function. Not at the same degree, but pretty big reduction, in a disproportionate reduction that you can see here in immunofluorescence. So we really think that this is at least in part mediated by IGF2R. And so the conclusions of all this work is that I think we now know why, what are the molecular mechanisms by which you have this growth of the placental vasculature. And this is induced by the fetal signal, the IGF2, endocrine IGF2, that comes along and can stimulate the growth in the endothelial cells, presumably through the IGF type 2 receptor, but it can also cross the endothelial cell, the fenestrated endothelial cells, and impact on the trophoblast morphogenesis directly. And of course, this work also shows that the endothelial cells themselves are a potent autocrine and paracrine molecule that acts on the trophoblast. And so I think there is a mixture of endocrine and paracrine effects to stabilize the normal growth of the placental vasculature. And this is how we think that the placental, the placenta growth is correlated to fetal growth is through IGF2 and IGF2R axis. And so these would be the second question, which I'm not going to do because I've clearly run out of time. Apologies for that. And I'm actually just going to go to the acknowledgement slides. You have seen already me acknowledging the collaborations, and these are actually the people that did most of the work. And again, apologies for overrunning. I will be very happy to take any questions that you may have.
Thank you very much, Miguel, for your talk, and also for sharing your data from your laboratory. I believe that this allows us to understand a little more for the complexities of the prenatal growth. So thank you very much. Unfortunately, we have time for a couple of questions. So
right.
It's Professor Chiarelli.
Miguel, thank you very much. I appreciated and enjoyed very much your lecture. Very strong data. A couple of points that I want to raise. We have observed a child with a lack of receptor for IGF-1, and this child was born very small, and he continued to grow very small. So IGF-1 receptor defects for pediatricians, as we know, although is a rare cause of short stature and intrauterine growth retardation, is still quite important for us as pediatricians. The second very point that I wanted to ask you directly is related to the IGF-2 receptor. You mentioned that the IGF-2 receptor through maybe also IGF-1 receptor through ERK, but may contribute to neuronal plasticity. This is quite fascinating issue, because we know that neuronal plasticity is very important during fetal life and continues during the couple of years after birth. So I would like you to speculate on that. And finally, a very short point, did you observe any difference in your model with the IGF-2 deletion if also other factors which can influence the placenta vessels, namely the VEGF, for instance, or other factors may contribute also to reduce the placenta?
Oh, thank you. Yeah. Great questions. So I'll actually pick up from the last one. So the VEGF is, of course, people know a lot about vasculogenesis and angiogenesis in the placenta, that's for sure. And they have identified all of these key regulators. VEGF, we didn't find is regulated at all. But the point I would like to make is the following. So this is an effect that happens very late in gestation. So most of these key molecules are completely essential for forming vessels. And this is, I think, the novelty of this is that this is a particular effect that acts very specifically in late gestation. And so we think there is this stimulation of the endothelial cell growth and proliferation and impact with the underlying trophoblast through the circulating IGF-2, but also endothelial IGF-2. So clearly, we don't think VEGF has a role in this. Probably surprisingly, because we thought we ought to see it, but probably this is a timing thing that this kicks in much earlier, the role of VEGF. So there is no evidence from any of the arrays that we've done. Now, in terms of your first question of the lack of IGF-1, so I'm not sure if so clearly IGF-1, like IGF-2, is very important in both prenatal growth. IGF-2 quite important in prenatal, probably not so much postnatally, at least in the mouse, but probably also in humans. We don't know for sure. But I think the surprise here was to see that the endothelial cells somehow don't seem to signal through IGF type 1 receptor, which is pretty fascinating in the sense that we don't think it's the insulin receptor because it's very lowly expressed. But we were surprised that it's not IGF-1 receptor. And so presumably what we need to do is endothelial specific deletion of IGF-1 But my guess is that if you take out IGF-1 from the endothelial cells, nothing much would happen, similarly to what we see for IGF-2. Your question about the neuronal plasticity, I think these are remarkable studies. But I have to say that people in the field, when you go against the dogma, and the dogma still is, no, IGF type 2 receptor does not have signaling properties, doesn't have catalytic activity, so it cannot possibly do that. From the perspective of the confirmation of the research. So we are really at loss to understand exactly how this, there's clearly from our work that is some IGF2 dependent actions, but probably some independent ones that go through the TGF pathway, which also binds the IGF type 2 receptor. So this is our next step, is to, so there's a mouse knockout that specifically targets the binding of IGF2, and we'll be able to, in the future, really try to tease that out, what is the role of the type 2 receptor in all of this. But I think it's fascinating that the endothelial cell seems to be different. And the placental endothelial cell, if you do a transcriptome, is probably going to be very distinct from of that to other fetal organs. Sorry for the convoluted answer.
No, but Alejandro, yeah. Miguel, yes. Can you hear me?
Yeah.
Okay, Miguel, this is Ceci Camacho Jimenez. Congratulations. Yeah. Beautiful, beautiful talk. I missed the first few minutes. I'm sorry, I was in another meeting, but
all right.
You know, you just answered my first question was the relation with the IGF2 receptor. As you said, for many of us in the field for a number of years, we have desperately tried to find out how is it working with the IGF2 receptor. And I'm looking forward to your next studies regarding this, because this is quite striking, actually, your data. My question is the second one, because that one you partially answer. The second one is that, you know, many years ago, I worked with the German group, and we did knockout of the IGF-1 and overexpression of IGF-2 using the mouse model. And we saw the major effect in kidneys, but it was not, we were not able to rescue the IGF-1 knockout with IGF-2 in the mouse, nor has been seen in humans when we have in patients with IGF-1 knockout gene, the IGF-2 circulating on this IGF-2 is quite high, and it doesn't rescue the severe short stature phenotype. So my question is, in your studies now with the IGF-2 knockout model that you have and the impressive data in placental growth that was suspected for many years and shown in different studies, but now is so clearly presented by your data. My question is, what happened postnatally when the IGF-2 in the mouse is severely downregulated? But if I remember one of your studies vaguely, you found that in postnatal mouse, IGF-2 still is found in some niche in the central nervous system. Maybe you presented that and I missed it because I was in the beginning of your talk. What is the story right now?
No, I haven't presented that.
Okay.
Thank you so much. Great, really great question. I'm not sure if I'll be able to answer properly, but my thoughts are the following. Yes, you're absolutely right. So IGF-2 in the mouse goes down dramatically postnatally. It's beautiful Spanish language being spoken.
I don't know from whom. Yeah, it's me.
So I was, so it's downregulated in most postnatal tissues, and there are exceptions.
Sorry, we had somebody interrupting the answer of Miguel. Sorry.
Yeah, so downregulation in most postnatal organs, exception being the brain. So there's the choroid plexus. Actually, IGF-2 is pretty much very well expressed and biallelic, so it's not imprinted at all, and also in the leptomeninges. And then, as you rightly say, it is definitely in stem cells of the neurons, progenitor cells, and also in the intestine, in the gut, in the S cells. And I guess all of those postnatal neuroplasticity that was mentioned before, it can have an action in the prenatal, but it can continue postnatally. I don't know if there are any studies specifically deleting IGF2 postnatally. And so this is what the field also needs, is to have deletions postnatally that take out the confounding effect of IGF2 programming many of these things through the actions in the prenatal life. So I guess your other point that I find fascinating as well is, why can't you actually rescue the IGF2 phenotypes with IGF1? I believe that's what you said, right? And I guess my answer is that IGF1, as far as we know, has very high activity through the IGF type 1 receptor and very little towards the other receptors. So IGF2 can do that, can signal through the insulin receptor, can signal through the hybrid insulin IGF type 1 receptors. And so it has a broader substrate of functions in terms of the receptor. And also probably is important is the timing of action between IGF1 and IGF2. That probably is not the same during prenatal life. I don't know. This is a great question.
And the last the last question, if I have, Ali, can I very quick question? Is you just mentioned the hybrid receptors. So for many years we also believe that hybrid receptors were found primarily in placenta. Is still that the case now of hybrid receptors, even if the IGF2 is not signaling through the hybrid? Or maybe, I don't know.
We don't have data on that. That's a very good question. No idea if we haven't specifically looked at the expression of those in, maybe something that we should be looking, because we already, I mean, we're excluding the IGF type 1 receptors being important, but it's still possible that it's through the hybrids. Although the only thing I would have against that is we don't see expression of the insulin receptor at all in the placenta.
I mean, that may be the answer. Because for years we were looking for hybrid receptors, and the people who were more into receptor biology always mentioned that the one of the organs that have hybrids was the placenta. So even though IGF2 from your studies working through IGF2 receptor, that is quite fascinating, or these are other binding to another receptor from IGF2 as a ligand. It is still interesting, the question that for a long time we have believed that placenta was the owner of the hybrids. So you have the beautiful model there, at least, to test it.
Thank you so much.
Okay, well, thank you very much for being here, especially to Miguel, to Cecilia, for Professor Querel to be with us today. So thank you very much, Miguel, and we really hope
thank you very much. It was a
great pleasure.
Again. Okay, thank you.
Thank you. Wonderful.
Bye, everyone. Stay well.
Bye.
Bye. Thank you.
Bye-bye.
Thank you. Bye.